Okay, so let's begin. Let me just begin by explaining the adaptive immune system. Okay, so here, so before the adaptive immune system or the third line of defense really starts, what actually activates the third line of defense are cells from the second line of defense, known as antigen presenting cells, which are two that we learn about in VCE Biology, which are dendritic cells and are also macrophages. So these both are phagocytes and they are also antigen presenting cells. They present antigens on their MHC2 marker to T helper cells. Okay, so let's begin. So when a pathogen comes into the body, it will breach the first line of defense, because we're going to talk about the adaptive immune system. So it will breach the first line of defense. Then it will breach the second line of defense, and then these macrophages and dendritic cells, they will phagocytose and then present the antigens of that pathogen on their MHC2 marker. Now the MHC2, when they present the antigen on their MHC2 marker, they will go and present that to a T helper cell that has a complementary T cell receptor. So, and this process of just antigen presentation is known as antigen presentation, and it's just MHC2 marker is presented to the T helper cell, to the complementary T helper cell that has. When we talk about complementary T helper cell, we're talking about the T cell receptor. So the T helper has a T cell receptor, and this receptor has to be complementary to the to the non-self antigen that's being presented on the MHC2 marker. So if the T helper cell is complementary, they will bind together, and once they bind, they click together. This dendritic cell or macrophage, so as we said, this is an antigen presenting cell, it will secrete cytokines, which will activate the T helper cell. Okay, once the T helper cell is activated, either the humoral immune response will be initiated or the cell mediated immune response. Okay, let's look at the different responses. So here we have the humoral response, which involves extracellular pathogens, and it involves B lymphocytes. So what happens is the T helper cell comes and it secretes cytokines to initiate or activate a specific B lymphocyte that will work to destroy that specific pathogen. After that, when this is activated, when this B lymphocyte is activated by the cytokines that are released by the T helper cell, it will undergo clonal expansion, where many copies of that B lymphocyte will be made. So many copies will be made, and then once it undergoes that clonal expansion, it will undergo clonal differentiation, where that B lymphocyte will differentiate into either B plasma cell or B memory cell. Now, most of the B lymphocytes will differentiate into B plasma cells, since they are the ones that do the work, and we don't really need too many B memory cells, because the body kind of doesn't need that much to remember that pathogen in the long term. So most of it is B plasma cell that the B lymphocytes are differentiated to. So we have clonal differentiation, where B plasma, B memory, the B memory cells will provide long-term memory, and B plasma cells will secrete and produce antibodies. Now, these antibodies have different functions, different purpose. They can do different things. They can neutralize, opsonize, immobilize, activate complement proteins, and agglutinate, which causes antigen-antibody complexes. Now, the way to remember it is think of piano. So P as in it activates a specific protein. So antibodies activate complement proteins. I, immobilization, A, agglutination, N, neutralization, and O, opsonization. Okay, so let's look at so that's the humoral response. Now let's look at the cell-mediated response. Now the cell-mediated response involves pathogens. So it is initiated when the pathogen is an intracellular pathogen, so a pathogen that infects the inside of a cell. So it involves T lymphocytes, and basically a T helper cell that is activated will come and secrete cytokines, just like it would in the humoral response. So it secretes, however, cytokines to a specific T lymphocyte this time. So it's T when we're talking about cell-mediated response. So yeah, it secretes cytokines to activate a specific T lymphocyte that will specifically work to destroy that specific pathogen, and then that T lymphocyte will undergo clonal expansion where many copies of it will be made, and then it will differentiate into, and then it will undergo clonal differentiation where it will differentiate into cytotoxic T cells and T memory cells. Now the T memory, just like B memory, they will provide long-term memory against that specific pathogen, and then the cytotoxic T cells will directly work to get rid of that pathogen, get rid of the cells that are infected by that pathogen by making them undergo apoptosis. So what happens is this cytotoxic T cell will go out and it will look for infected cells by that pathogen. Say it was a virus, a specific type of virus. Once it finds it, it will secrete death ligands, and these death ligands will cause apoptosis to occur, which is also known as programmed cell death. And the cell will basically turn into little fragments, and it will be basically destroyed into debris, so waste. And yeah, so that is the two different responses that occur at the adaptive immune system, and depending on the pathogen and the nature of the pathogen, either the human response or the cell-mediated immune response will occur. However, both of them could also occur. But yeah, I hope this video really helped you out and cleared that information about the adaptive immune system and gave you more insights. And it's a short video, but I hope it helped in some way, and thank you for listening.